Let us talk about cilia and flagella now. We are taking both these structures together because their structure, basically the structural makeup is same, but before that we should see the differences between the two. Cilia are normally very small, thin, hairy structures. Small, thin and hairy structures. Hairy in the sense their appearance is like hairy. So hairy structures. Flagella are long. Normally their base is wide and the tip gets narrower. So they are long, we can say whip-like or hunter-like structures. Whip-like structures. A cell invariably has more number of cilia. We will talk about the types where there is only one cilium present, but normally their number is more. So they are numerous. The number is invariably more per cell. And flagella are normally 1 to few per cell. 1 to few per cell. Cilia normally move together. That means in the form of some kind of a harmony. So their movements are harmonious. All cilia would move together. They can move in a synchronized manner. So movement could be synchronized. Synchronized is, suppose there are all these 4 cilia and all of them move together, then that movement would be called synchronized movement. So the movements are synchronized, that means all move together. Or it could be a rhythmic movement, that means if same 4 cilia, this one moves first, then next, then next, then next. So it's a sequential movement. So it could be rhythmic where all of them move, but first one moves, then the second one moves, then the third one. So it creates a wave kind of a thing. Whereas in case of flagellum, it shows undulating movements. Undulating movement. Undulating movement is in the form of a wave. So one structure itself moves with ups and downs, folds, which is known as undulating movements. In certain ciliates, many cilia have fused to form one structure. So if cilia fuse to form that one structure, it is known as cirrus or cirri. It is found in very few ciliates. Flagella, normally they don't fuse, but sometimes if this is a flagellum, then, and this is the organism, so it would have a membrane between the flagellum and the body. It would just help in the movement of the organism. So they don't fuse with flagella, but it can have a membrane connection with the cell. Structure we will be talking about very soon. Types of cilia. There are 2 types. One is called kinocilia. So one is kinocilia and other is stereocilia. Kinocilia are motile cilia. That means they move, they are motile. And stereocilia, they are non-motile, they don't move. So they are non-motile. Kinocilia are the ones which we were talking about, which help in locomotion or movement of substances from one place to another. Stereocilia are non-motile and they are found in sensory cells. Sensory cells like our taste buds, those cilia, the receptors which we have in our ears, they also have these kind of cilia. So they are non-motile. Their function is not to help in transport of something. It is just one place. If it gets stimulated, it will pass that stimulus to the neuron, through the neurons to the brain. So two types. Flagella are also of two types. These two types are on the basis of their structure, shape, external. One is known as tinsel type, and other is known as whiplash. Tinsel is broad from the base and goes on becoming narrower, plus it has some stiff hair-like structures. Whereas in case of whiplash, which is a typical hunter-shaped structure, the base is thicker as compared to the tip, but it uniformly gets tapering towards the tip, and there are no such bristles or hairy structures on the whiplash type. So two types again on the basis of external structure. The cells where we find cilia and flagella. Cilia are present in all ciliates like paramecium, they are all over the body. Cilia are also present in the kidney tubules where they have to conduct urine. Cilia are also present in the fallopian tube where again conduction of egg and then fertilized egg, that is zygote, takes place. Flagella are present normally in cells which have to move from one place to another, like zoospores. Zoospores have flagella, zoospores of Chlamydomonas or Spirogyra, they have flagella because they have to move from the male reproductive structure to the female reproductive structure for fertilization. So flagellate cells, plus Euglena, Trypanosoma, they are also flagellates. Now let us see the structure, internal structure. When we were talking about centriole and basal granule, we said the arrangement is 9 plus 0. Here the arrangement is 9 plus 2. That means 9 microtubules make the peripheral part and 2 in the center. Nine peripheral, made up of microtubules, same type of structure, but instead of a triplet, it is a doublet. So there are two things, the microtubules are in the form of doublet. There is one unit which is called A and the other unit is B. So this is A and B. We can also call them alpha and beta. A is made up of 13 microtubules, 8, 9, 10, 11, 12 and 13. And B is C-shaped. It is made up of 10. So 13 here and 10 here. These doublets would make the 9 peripheral ones. Let us make the A's first and then we will draw the B's. Nine, only A's we have drawn. The B is slightly bigger, wider. as you can see, the A is slightly inner and B is slightly outer. Now, from A arise two arms. One arm is straight and other arm is bent. Each A has these two arms. So, inner arm and outer arm, which is bent, inner and outer. So, from A arise two arms. These two things are made up of tubulin protein. These arms, inner and outer arm, they are made up of dynein protein. This is the outer arm and arm. Both are made up of dynein protein, and dynein protein acts as ATPase. That means it will hydrolyze ATP to get energy, and that energy will be used for the movement of these cilia and flagella. We said the arrangement is 9 plus 2. We have made the 9 outer and we have seen what these A and B are made up of. Microtubules 13 in A, 10 in B. Each A has two arms, an inner arm and outer arm. The outer arm is folded. Let us come to these central two. So, there are two microtubules in the middle, and these two are connected by two cross bridges. These are also made up of tubulin protein. And these cross bridges. This is the central microtubule. The central two microtubules are surrounded by a membrane. So, here there is a membrane which surrounds these two. In case of centriole, in the center there was a hub, no microtubules, plus there was nothing of this kind of arrangement. From each A arise a radial spoke. So, the radial spoke is again a filament or a fiber. It comes up to the membrane of the central core and it has a thickening here. This thickening is known as foot. So, let us draw all 9 first and then we will label. See, it is arising from A, it is not touching the membrane and there is a thickening very close to the membrane of the central microtubules. So, these are radial spokes and the swollen structure which we find at the base of each radial spoke is known as a foot, which is just a swelling of the protein. Some more connections. The outer arm is connected to B of next. So, let us name these. This is A, B, this is A and B. So, outer arm connected to B. So, from here there is a connection. Again, outer arm to B is a connection. Outer arm to B. Outer to B. Again, these connections are going to help in letting it move in the form of one unit. These connections can be termed as AB connections. So this was A, this is B, this is again A and B. So these connections are helping in A, arm of A and B. So we can call them AB connections. They can also be termed as just interconnections, and they are made up of a protein called nexin. So now there are three proteins which are found in this: tubulin, dynein. So one is this tubulin protein, which is the major constituent of the microtubule, dynein protein, and the third one is this nexin protein. So these three important proteins are present here. These two central microtubules, they are known as C1 and C2. How do we differentiate C1 and C2? Is a very simple thing. From C1, there are some fibers which radiate. These fibers are also made up of the same protein, nexin. So these are fibers of C1. So if only two are drawn without any labeling or anything and one has fibers, you can easily conclude that the one with fiber is C1 and the other one is C2. So there are two on in the center part. This complete structure is surrounded by a membrane, the typical plasma membrane. In case of centriole and basal granule, there was no membrane cover around it. So there is a membrane around this. And it is similar to plasma membrane. So cilium or flagellum, they are membrane bound structures, whereas centriole and basal granules are not. There is no membrane which covers them. Both cilia and flagella have similar structures. These proteins which are here, they use ATP to generate energy. Once that ATP is hydrolyzed, then that ATP will help in connection of this foot to the membrane. When the foot part of all these 9 spokes anchors or connects the central part, the complete structure would move as one unit. So with expenditure of energy, these foot parts would connect to the membrane and then the movement will take place. This is the structure of a typical eukaryotic cilia and flagella. Bacterial flagella have a different structure. We will talk about that in some time. But structurally, all are microtubular structures: centriole, basal granule, cilia and flagella. But two, that is centriole and basal granule, they have 9+0 arrangement. There is nothing in the center. Cilia and flagella have 9+2 arrangement. One more difference is that centriole and basal granule do not have this external membrane sheath, whereas cilia and flagella have this membrane. Now let us talk about the flagellum and the functions. Cilia, flagella, basically for conduction of certain things or for capturing food or for locomotion, especially cilia, flagella, locomotive structures. Now let us take the structure of a prokaryotic flagellum. Prokaryotic flagellum is not a complex structure like this. Its structure is much different than that. So let us talk about that structure. So now let us take the prokaryotic flagellum. Flagella of prokaryotes or prokaryotic cells. We just now saw the structure of a typical eukaryotic cilium and flagellum, 9+2 arrangement. Here the flagella are made up of a protein called flagellin. And the flagella has two parts, a base, or which is also known as foot, and filament. The base part again has two parts, the shaft and hook, and then filament. So basically speaking, there are three parts. There is a shaft, there is a hook, and there is a filament. If we see a flagellum, the shaft part, it comprises of discs, and these discs, they are connected to each other. So this is one structure, then again there is an extension and two more. So there are four discs which make up the shaft in a gram-negative bacterium. The lower disc is connected to plasma membrane. And the outer disc is in the cell wall. So this is connected to the peptidoglycan cell wall. Connected to this is the hook part, and then extending from the hook is this flagellum. So there are three parts which we talk of. This is the shaft, the hook, and this is the filament part. This is seen in case of gram-negative bacteria. In gram-positive, everything is same except for one difference, that instead of 4 discs, there are only 2 discs. So there would be one disc which is connected to the plasma membrane. This will be to the plasma membrane, and the outer one will be connected to the cell wall. And then there is a hook which is again same as what is seen in case of a negative bacterium, and a filament. So this would be the shaft, hook, and filament. And the protein which makes, let us write this as gram-positive bacteria. So the protein which makes the flagellum of prokaryotes is made up of a simple protein flagellin. We don't see 9+2 arrangement. The difference in the structure is that the shaft of negative bacterium has 4 discs, and the shaft of positive, gram-positive, has only 2 discs. The lowest disc or the innermost disc is attached to the plasma membrane, and the outermost is attached to the cell wall. Same is the case in case of the gram-positive also. Movement is again with the expenditure of energy, and because of this shaft and hook, the flagellum is able to move 360 degrees. So it is just an anchoring thing, and this whole structure can rotate completely 360 degrees. So simple structure as compared to eukaryotic flagellum.